Hi everyone, welcome to the Research Like a Pro with DNA question and answer series. I'm Nicole Dyer and today's video is about the DNA matrix tool at DNA Painter, and this is an excerpt from last month's office hours in June when Johnny Pearl released a new tool called DNA Matrix Tool. And so I'm just kind of going over what the tool can do, giving a couple examples of how you might use it. And since I made this video, Johnny has done some updates to the tool, so I will link to that in the description below the video so you can see the updates. But basically now there are some more options for visualization within the tool, including changing the colors, and he has also added a way to view the standard deviation as well. So that can help flag relationships that are a little bit higher than one standard deviation above or below the mean to help you flag anyone that's a kind of outside what you would expect for that relationship, but still within the range, the full range for the shared Sun Morgan project data. So it will be helpful to help you visualize people and how much they share with each other, as well as flagging any potential outliers. I hope you enjoy this video and learn more about how you can use the DNA Matrix tool, and be sure to read Johnny Pearl's blog posts about the tool as well and watch his webinar at Legacy Family Tree Webinars. It's the new DNA Matrix tool at DNA Painter. And when I see a tool like this, I definitely feel like I'm seeing a genetic network and seeing how much they share with each other. If you can just kind of imagine a cluster matrix chart and think of this as just one of the clusters where all these people share DNA with each other, and we don't usually see the amounts that they share with each other in those cluster matrix charts. We just see that they do share with each other. For like, we'll see a colored cell in between Nathan and RG. We'll see that they share, but it won't always say how much. But this tool allows us to put in the actual amount they share and their relationship, and then it can compare it against the shared Sun Morgan project. So Johnny introduced this tool last month in May, and he gave a webinar for Legacy Family Tree Webinars that was really helpful. And so I recommend that you watch that if you can, to kind of think about and brainstorm how it might be useful to you. Johnny shares why he wanted to create it and what he was going to use it for, which was sharing this information with his close relatives. So it focuses on the people who he's tested in his family, including his close relatives like parents and siblings and nieces and that kind of thing. The tool is brand new. It's kind of in beta, he says in the webinar. He has a blog post introducing it, and he plans on rolling out some more features to it. And he is curious how people will use it to help them with their research questions. And since he created it to kind of be more as a tool for sharing and collaborating with relatives, and like a presentation tool, like, here, look at this, and you can send it to them. But he's aware that other people will probably want to use it for other uses, like solving research questions. So if you think of something that some functionality that would help with that, we can send it to Johnny. So there are the options to show centimorgan or to show the percent of DNA shared. And there's also two shading options right now, which he plans to add more colors and things to choose from. But right now, the default is that it is colored based on the DNA shared versus, and then you can switch over to what's expected based on the shared centimorgan project, and then it will colorize it that way. There are two ways to enter data. You can do manual entry where you type in the basic tester info into a profile for them with their name, initials, platform if you want, kit, and notes. You only need to do the name, then the initial populate for you, and the rest are optional. Then you can go into edit mode, and so you can see after you enter all the testers, you can see the whole grid and you can just start typing in numbers. Or you can open an individual tester and go to the tab for entering all their amounts. If that sounds like a lot of manual data entry, then if you already have one of these things, then you can import the data. So maybe you already have a spreadsheet that has a matrix. It has to be a matrix that looks like the matrix that he has here. If you have a spreadsheet that looks like this with the same on the side and on the top, then you can import that spreadsheet. Or you can go to GEDMatch, and GEDMatch has a matrix tool that you can generate, and then you can copy and paste that into DNA Painter's matrix tool. FamilyTreeDNA does have a matrix too, but it's limited to 10. And it would be challenging to not challenging, but you could put in more people than just 10, but it would be more manual data entry. But you could do it. But you can, it's something to try if you just want to play with the tool to go into the shared matches are in common with for one of your people in your family tree DNA matches, and then create a matrix with 10 of their shared matches, and then put that into DNA Painter and put in some of the relationships that you think there are. So the relationships have to be added manually. So there's right now there's no way to import those, but that will probably be a new feature eventually. So you do have to type those in yourself. Okay, so I mentioned two types of shading. So the default is that it has DNA shared selected, so that the darker the cells are, that means they're closer genetic relatives. And the lighter the cells are, that means they're more distant relatives. So you can see the darkest ones are like these parent-child ones right here. And then if you switch it over to the DNA shared versus expected, so switch over to the versus expected, then matches that are in the 96, so for example, a high percentile, like 96th percentile, is going to be green. So I'd like to think about that as like the green light. I have to think of these things to help me remember what the colors mean. Because right now you can't change the colors, but maybe in the future. So if it's a really close match to what's expected for the relationship, it'll be green, like a green light, which means go. So it's good. If it's less close to what you would expect, then it's going to be purple. So if it's a low percentile, like the 5th percentile will be purple. So you can see that a bunch of these relationships are green, but some of them are more purplish, meaning maybe they aren't as common amounts for that relationship. Another thing you can do is reorder the columns, but it's not able to like drag and drop yet, but you can click on one of the people in the top columns and then it will order by shared DNA with that person. So clicking on Amanda will put her in the first position, and then everyone else will be ordered according to the type of shading that you have selected at the moment. So if you have the DNA shared, then those who share the most will appear first. If you have the versus expected selected, those who share in the highest percentile will appear first. So I was trying to brainstorm possible uses and think about like what can we do with this tool. So some of the things I came up with were to compare hypothesized relationships with the shared Centimorgan project to see if they're kind of matching up and to kind of do a lot of comparisons at once. You could do this in a couple ways I thought of. Let's go back. One is to compare GEDMatch and FamilyTreeDNA clusters with the shared Centimorgan project data. So let's say you have a cluster in GEDMatch and you put it into a matrix, then you can copy and paste that matrix into the matrix tool, and then you can check all that data against the shared Centimorgan project data all at once. Another thing to do would be to go to the Ancestry Pro Tools shared matches. Maybe you have a cluster that you've given a colored dot, and you can even focus in on just those with a common ancestor hint, because then it will give you a hypothesized relationship that you can test. Then you can manually put that in and test it with the shared Centimorgan project data. And this kind of thing can help you identify any outliers or any problem matches where the data isn't lining up with the hypothesis. And then, of course, the use that this was created for was reporting and collaboration, where Johnny wanted to share a matrix with his close relatives so they could see how much they all share with each other. And this can be used in many different ways, maybe create a visual aid for sharing in a report. This kind of table would be really useful. It's succinct. It could fit within an 8.5 by 11 page, which sometimes is a challenge. And the way that the matrix has just initials along the top and the full name along the side makes it easier to fit on a page. So you could do this for like close matches of a test taker towards the beginning of the report to show all these people are siblings, first cousins, and sharing in expected amounts before you dive deeper into the DNA analysis. Another thing is that you may want to show how much everyone shares both in centimorgans and in percentages. So this is a really easy way to convert a lot of shared DNA into percentages to help the readers understand, because most people who aren't genetic genealogists don't understand how much DNA is 150 centimorgans. And then if you're going to be emailing or messaging relatives, you can send them a link to the matrix or generate an image of the matrix to share with them. So a lot of possible uses, and I think there will be more cropping up. One thing that Johnny mentioned in his webinar is that Banyan DNA takes this type of data as input and then allows you to verify it with verification scores, but it currently doesn't have a way of showing a matrix of all that data. So it's interesting that that's not available there. And when he mentioned that, I wondered if there would be a possible future collaboration there. But it is good to have the matrix data, and I've used matrices in the past that I've made on my own in Google Sheets. Sometimes it just feels like you need to view things in a matrix. So here are two examples. And so this first example was just me trying to play with the tool and see what it can do and learn how to use it. And so I recommend that everyone try doing this too. Whenever you want to try a new tool, just try to click around and start typing things in and see what happens. You are allowed to create one free DNA matrix, and then if you want to create multiple, then you'll need to subscribe, just like other tools on DNA Painter. So let's start with what my goal was. So I'm working on my Sally Keaton research project with the objective of figuring out if Sally Keaton was the sister of Lucinda Keaton. Both Sally and Lucinda were mentioned in their father, William Keaton's estate proceedings in 1830 in Anderson District, South Carolina. And right now I have multiple test takers who descend from William Keaton's other lines, mostly from Lucinda's descendancy, and some descending from other of William Keaton's descendants. But I wanted to see if I could find matches to Sally's descendants. So the first thing that you do when you're creating a matrix is add a title. So my title was William Keaton Descendants. And then the next thing you do is just click add a tester. Now, one thing I did first was I did play with Johnny's example. So if you want to just not have to put any data in, but see how it works, you can click on his example data, and that allows you to click on things, open it up, read the information, and see how it works. But when you're ready to add your own data, click add tester, and then this window will pop up and you can type in the name. So I typed in a privatized or anonymized name for one of my test takers. I put in Lucinda 1, and then it automatically put in the initials for me. And then I did that for two more test takers, Lucinda 2 and Sally 1. And then I did a couple more. And then the next thing you can do after you've added all your test takers is go to Relatives and Shared DNA on that same tab where you added the profile. And once you click that, you'll see that for Sally 1, I can now add how much she shares with all the other test takers, and you can put in their relationship. So Sally 1 didn't match Lucinda 1, but she did match Lucinda 2, Lucinda 3, and Jessie 1. So Lucinda, Jessie, and Sally are all descendants of William Keaton. So these are through different child lines. So I put in the amount of shared DNA and the relationship. What's great about typing into the relationship is that you can type it in however you normally do. If you type out the word fourth, F-O-U-R-T-H, or type in 4C2R, it will figure it out and then convert it to this standardized format for you. Now, after I added in all the shared DNA, then I had this matrix, and I didn't complete all of it, and some people didn't share. Actually, these people didn't share with each other. And one thing to note about reading a matrix is that the top half is identical to the bottom half, which kind of is redundant and isn't as efficient. But because you don't have to type it in both spots like you normally would if you're making your own matrix, it's probably better to use this tool. One way to remove that redundancy if you're making your own is to just not put in the top part and just leave this blank. I think it would be less confusing for readers also to not have to see the info twice and wonder why is it in there twice. But the output looks like this. I like that it has the names along the left fully spelled out and then the initials along the top. And here is what it looks like when I converted it to percentages. And then here's what it looks like when I converted it to back to centimorgans, but then changed it to versus expected, which is going to compare it against the shared centimorgan project data. So now you can see that most of these amounts are in the lower percentiles for the relationship, except for this one, which is in the middle. So I don't have any that are in the highest percentile. The next thing I played with was looking at the export options. So if you go over to the side and you click export or share, this is what you see. So there's actually a lot of ways to export, and you can duplicate the matrix if you want another copy of it to make anonymous for some reason or do something else with it. Or you can save an image of this, which would be great for just downloading it to your computer to put into a research report or sending in an email. You can also export it as a CSV to put into Microsoft Excel, or export as a JSON file so that you can load it back into the tool later, which is a great option if you are wanting to use the free plan. When you click share, it says the matrix is private, but if you click share, then it opens up this and it gives you a link. And then you can click shorten the names for privacy, and then it only affects the link that you're sharing so that when you're working on it, you can see the full names, but when other people see it, they will only see the anonymized view. Well, I thought this was a bit cumbersome typing everyone's name and data into the tool, especially when I usually have already tracked this data in my Airtable log. So I was trying to think of a usage for this that wouldn't be cumbersome or that wouldn't be repetitive. And I thought of an idea that I thought I would just try for today, just to learn the tool. So what I thought about was using AI to help get the data into a matrix. Then I could put it into the matrix tool to compare it against the shared centimorgan data. So that's definitely a strength of the tool, is that it can compare it with the shared centimorgan data and help you find outliers and help you notice any problems with your data. So I decided to use Claude because of its strong privacy settings. The data is not fed back into the model unless you give it a thumbs up or a thumbs down. So it's more private. So I have the same objective with this one. I'm still working with the Keaton descendants, and I'm focusing on one of my test takers, John, who he's a close, generationally close descendant of the Keatons. And I focused in on his matches that were identified as part of the Keatons in Ancestry, the Keaton cluster. And my goal was to be able to get AI to generate a matrix that I could then paste right here, because in the matrix tool, there's a button for load. So if you click load, you can then paste information right here. Just paste the matrix and then it will automatically populate in the tool for you. So here's how I did it. I went to Ancestry. I filtered to John's parent 2, which is the right line, right side. And then I chose one of the matches who I had labeled as a Keaton match. And then I went to the shared matches for that person. And so that was Lois. And then I looked at Lois's shared matches with John, and I have the Pro Tool so I can see how much they all share with John and with Lois. And then I wanted a smaller set of matches to work with, so I filtered to people with common ancestor hints so that I would be able to put in the relationship with John. And so then I took this screenshot and I pasted this screenshot into Claude and said, help me create a DNA matrix using this information. 6 DNA testers on the vertical axis are showing how much they share with John and Lois. The matrix should show the amount of centimorgans John and Lois share with each of these testers. So I used Claude Sonnet 4, and it created a very fancy matrix that was a little bit more than I wanted, but it was pretty nice. So that was good. I kept adding more data into this by just chatting to Claude and saying, well, Mary shares blah, blah, blah with so-and-so, and David and Jeremiah share this much. And so as I added the data, it added additional columns for how much Amanda shared with everyone else, how much David shared with everyone else. And I did notice a pattern that Mary shared with no one else except for John and Lois. And that was interesting. As I added more data, Claude created multiple versions of the matrix. I finally asked for a simplified version that included all the names along the top and the side. So then I finally got the kind of matrix that I was thinking of, just like this. I asked it, what data am I missing? And then I found that I was missing like one of Todd's matches, and then Mary just didn't match anyone else except for John and Lois. So when I looked at Mary's common ancestors with John, I noticed why she's not sharing with the others. She was on a different line, but it was um, she shared DNA with Lois because Lois also shared a line with her, but not with these others who are in the Keaton cluster. So she wasn't really in the Keaton cluster. Okay, so now that I had this data into a matrix, which was great because I didn't have to type everything out. It wasn't as cumbersome. It was kind of a co-worker with me where it was typing things in and I was finding the data that was needed and feeding it in. And then I could just go right here. I literally selected this with my cursor, did Control C to copy this whole matrix, and then went right here and did Control V. And as soon as I did that, this was populated. So it was very immediate. It almost surprised me. And so that was great. And I did check that to make sure it was the same. And so then I went into John's profile. So the way I did that was by clicking on John's name, and then it opens up his profile, and I clicked edit. Once I clicked edit, I went to the tab relatives and shared DNA. And that's where I went and entered all the relationships of Jeremiah, Amanda, Todd, Mary, Lois, David, and Brian. And remember, I selected them because they all had common ancestor hints with John. That's how I filtered the shared matches list to use. So then I inputted all of their relationships based on the common ancestor hint. Then I clicked save, and then I went back and changed it to compare it against the shared Centimorgan project by clicking versus expected. And that's when I saw this big red cell between Brian and John. So the only parts that are colored here are the relationships between John and the other test takers because I didn't look at the results of any of the others. I don't have their tests. I just have John's test. I know how much these people share with each other, but I can't get common ancestor hints about how much they share. I can only go figure out by looking at their trees and comparing and figuring out. So I could do that manually, but I didn't do it this time because I was just doing a quick test. And it was a good test because it showed the value of this tool for pointing out outliers. And so this match between Brian and John was high. It's actually out of range for first cousin 3 times removed, so it's too high for that relationship. And if you click View in Shared Centimorgan Project right here, then it opens up the histogram for first cousins 3 times removed. And the amount I entered was 281, and this is outside the recorded bounds, 99th percentile for first cousin 3 times removed in the Shared Centimorgan Project. The highest was under 275. It was within the 250 to 275 bin, and so it's outside of the range. So what does that tell me? Now what do I do? Well, I just need to go back to the match between John and Brian, look at the tree, look at the shared matches, and see if there's anything fishy going on with this match. My hypothesis is that this match is either not a first cousin 3 times removed, maybe it's only 2 times removed, or maybe they share multiple common ancestral couples. So looking here, I can see I've actually given them, Brian, 2 clusters, the Bone and Harris line, which is the ancestral couple between John and Brian along the descent line of the Keatons. And then this other side of John's family that's not related to me, Kitchens and Punch. So it appears these two are sharing in 2 ways along 2 lines. So this was such a great tool for highlighting inconsistencies within a cluster. It was neat to try using Claude AI along with it to see if I could speed up the process of putting in the data. It would be fun to also try taking a FamilyTreeDNA matches matrix and putting that in, as well as importing a GEDMatch cluster. And it will be great to follow along with this tool and see new features that are added along the way. Okay, so are we having any comments or questions about the DNA matrix tool?
Yeah, we do have a few that we can talk about. So the first question is it okay to combine DNA data from multiple testing companies in one matrix?
Yeah, you can definitely try doing that. The question is, will you have any data overlapping? So if you have all the same test takers in one company that you do in another company, then you'll be able to compare. But if like John is at Ancestry and Brian is at 23andMe, you won't know how much they share unless you compare them at GEDMatch. So of course you can try to put them in, but the goal of the matrix is to compare multiple people to multiple other people who all have shared amounts. And with this example, let's see, there were only a few zeros here at the bottom with Mary, and then there was this one zero between Todd and David. But the rest were all at Ancestry, so I could see how much they shared with each other because of Pro Tools shared matches. Otherwise, like if one of these or two of these were at 23andMe, then I would be able to put in how much they share with each other. So I could fill out like one of the cells between Todd and Brian, like this one, Todd and Brian. I could fill out that cell, but the other ones I would have to put no data because Todd is at 23andMe and Jeremiah is at Ancestry, so I don't know how much they share. Does that make sense?
Yeah, it does. And it seems like it would just get confusing because you'd see those zeros and there's really no place to put there which company you tested at. Or wait, maybe I missed that. Is there a
Yeah,
place? It
there
does
is.
show. Okay. So just like on the chromosome map, you can put where people test. So yeah, so you could put that then by just clicking on the names, you'd see right where they tested.
Yeah, but it wouldn't be that useful. So the whole point of what I was
Yeah,
saying is that there's no reason to do that because you don't have data to compare those
right,
people. The point
right.
of it is to compare how much these people share with multiple other people in the same
Mm-hmm.
cluster. So you want them to all be from the same company. And then you can just make another cluster for the other company. It's like the
Yeah.
leads method. You have to do the leads method for each company
Mm-hmm.
separate because shared matches do not overlap. You can't figure out how much somebody at 23andMe shares with all these other cluster people over here. So overlapping with other testing companies is only able to be done with segment data or if you compare at GEDmatch.
Perfect. Thank you. Okay, and then another question. How do you get common ancestor hints? I have Pro Tools and not seen that feature.
You actually don't need Pro Tools for that feature. It's just that um, you anyone has that. Let me go back. It's right here. If you have this symbol, let me see if I can zoom in. If you have this little symbol that has the people, the person next to it, that means that Ancestry has compared your tree with the DNA matches tree and they think they know who the common ancestor is. So when you go to the DNA match page, it will have, we think you are first cousins 3 times removed and your common ancestors are Francis Marion Bone and Melissa Margaret Harris.
Perfect. Thanks for showing that. And one caveat with that is if you've got some pedigree collapse or double cousins, it will only show you one set of ancestors. It'll only show you one couple. At least I haven't seen it showing multiple couples when I know there's two couples that we share.
Well, it used to,
Have you
and I don't
seen
know
that?
if that went away or not, but
Ah,
it sometimes does and sometimes doesn't. And if it does find more than one common ancestral couple, at the bottom of the common ancestor hint, you'll see a link that says view other common ancestors. And then it will
interesting.
show multiple. But with this example, with the first cousin 3 times removed, it didn't show it right here. Or no, this was the 2 times removed. Anyway, one of these, it didn't show the other common ancestral
Mm-hmm.
couple. I just saw the two clusters that I had assigned it to and didn't compare the trees because I ran out of time. But yeah, sometimes it will show and sometimes it won't.
Good to know. All right, that is it for our questions. That was really interesting. I especially like how you used AI to help you fill out all the information and do the matrix. That was awesome.
Yeah, honestly, as I was doing that, I was thinking, like, what is the value of putting this into the
Hmm.
matrix tool at this point? Because I can just have AI do it all for me over here. And I could probably just upload a PDF of the shared Centimorgan project data and have it do the same thing that the matrix tool was doing.
Mm-hmm.
So that was interesting as well. Um,
I guess the one good thing about the DNA Painter matrix is you can click on it and see the little, it gives you a little bit different kind of histogram, but you do get to see another visual,
mhm.
as you're looking through it.
Yeah, that was definitely my main takeaway was that the connection to the shared Morgan project data was really nice.
Hmm.
And just knowing that it's compared against that accurate data set with AI, you're always having to validate and question everything. So
Yeah.
having it generate the matrix first and then getting it out of there and putting it into a trusted tool where you can have it evaluate the amount would be great. I did have to type in all the relationships, but it wasn't that bad.
Yeah. Well, with DNA work, there is always a certain amount of that typing in stuff. There's a little bit of busy work sometimes that comes with it. But yeah, this is a neat tool. I think it'll be fun to see what we can do. And I actually really like the idea of using it for a report because I think it'd be such a great visual. Often we're just doing a table, and this might be kind of neat. I wish there was a way to hide half of it, though. Like you were saying, it gets confusing for people that aren't used to it.
Well, I actually figured that out with AI. You can just take the matrix and put it into AI and say, make this into a triangle matrix.
Okay, there you go.